Cladistics is a way of grouping organisms together so that we can see the ancestor-descendant relationships. And when we look at these relationships, the idea here is that we're going to place everything into groups which we call clades. So when we look at those clades, we're going to be able to see what descendants came from each common ancestor. The overall picture that we come up with would be something similar to what you see on this slide. This is what we would call a cladogram. Notice that we've got various branches in this tree, and at each branch point, that would be a common ancestor. The groupings on this tree can represent different types of clades. And if we go through those types of clades, the first one is what we call a monophyletic. This is called mono, and mono stands for one, and this is because we have one common ancestor that gave rise to all of the descendants that we see here. So that one common ancestor would be right back here, and then notice that this clade is going to include all of the descendants from that ancestor. Other types of clades that we can have paraphyletic. Paraphyletic has the ancestor, but then notice that we only have part of the descendants. So we're missing some of the descendants in this clade. This is not ideal. When we're talking about clades, we really want the clade to have the common ancestor, but then we also want it to include all of the descendants from that ancestor. Another type is the polyphyletic. Polyphyletic has the descendants, but it's missing the other aspect. It's missing that common ancestor. Notice that the common ancestor for these is located outside of the clade. Okay, so what this means to us is that the polyphyletic and the paraphyletic are not really what we would consider true clades because they're missing the two important aspects. We want the clade to have the common ancestor, and then number two, we want it to have all the descendants. So if it doesn't meet these two characteristics, then we're not going to consider it a true clade, and it's not completely useful for determining the ancestor-descendant relationships. Here's a look at a cladogram for some dinosaurs. And if we just go through the different types of clades again, this would be what we would call a monophyletic clade. So this is a good clade. It's useful for cladistics. We can see that way back here is where we would have our common ancestor for these many species that we're looking at. This one here would be the paraphyletic. Notice that we've got the ancestor, but we're missing a huge number of the descendants from this ancestor. So this is not going to be a clade that we want to use. And then this over here would be the polyphyletic. We've got the descendants, but we're missing that common ancestor. For this one, the common ancestor will be way back here. So again, that's not one that we want to use for all of this. We want to stick with the monophyletic ones, the ones that have the common ancestor, but then the ones that also include all of the descendants from that ancestor. So as we put together these cladograms, what we want to think about are things very similar to what we were talking about when we put together phylogenetic trees. We want to look at similarities. When we start to look at similarities, just as before, it's very important that we're able to determine which similarities are analogous and which ones are homologous. The analogous ones are not useful to us because they don't have anything to do with a common evolutionary history. The homologies, though, these are ones that are due to a common ancestor. So those are the ones that are going to be useful. We want to try to eliminate analogous characters and really focus in on the homologous ones. When we look at the homologous ones, we can divide them into two categories. We have what we call primitive ones and derived ones. The primitive hopefully sounds old to you. That's what that means. And this is something that is going to be found in many groups. And then the derived characters, these are ones that are going to be unique to a clade. These are the ones that help us to really start to determine the pattern or the timing of the descendants from that particular common ancestor. If we look at how all of this fits together, one of the easiest or most common ways that this branching and grouping is determined is using outgrouping comparison. With outgrouping comparison, we're starting to distinguish between those primitive characters versus the derived characters. So what we would do in this case is we would have a number of organisms that we want to determine their relationship with each other. So these are ones that we are assuming all belong to the same clade. We also have a number of characters, and these are going to be homologous characters that we're going to be comparing. When we put this together, it's called an outgrouping comparison because we identify one species that is very different from the others. And this is what we call our outgroup. So by different, it's not as related to them as they are related, the rest of them are related to each other. So this is our outgroup. On this table here, we've indicated whether these various species have a trait present or whether it's absent. So A is for absent, and P is going to be for present. So we've got a nice organized chart here that we can refer to as we start to put together our cladogram. So when we start the cladogram, we have to keep in mind that all members of the clade must share at least one derived characteristic. So we need at least one characteristic that is different from that all of them have, that is going to be different from that outgroup. We'll start by making the two branches of our tree, and we put on the two sides the outgroup, and then on the other far side we want to have the most different individual or organism, which in this case is going to be human. So if you look at the way all of the characters played out in our little table, we've got the outgroup at the top and we've got the human at the bottom because those two were so different from each other in all of the characteristics that we're looking at. Once we get past that character that's going to be present in all of them, we would want to identify a character that's going to be present only in the members of this clade. And in this case, it's going to be the vertebrae. Notice that every one of them has the vertebrae with the exception of the outgroup. So this would be where they start to differ from each other. We put the hagfish on the cladogram because the hagfish has this vertebrae present, but it's missing everything else that we want to look at in this case. The next distinguishing derived character is going to be the jaws. We have the sunfish separating off here because the sunfish has the jaws, but it doesn't have any of the other characters that we're looking at. And we would simply go through our table one by one, constructing this cladogram. So what we're doing is we're showing where the common ancestors were and what derived characteristics that common ancestor had. So if we looked at this, if we, for instance, just looked at this group here, we could see that all of these four species that we have, or four groups that we have, they have this common ancestor and they're all going to have the common amniotic egg. This would not be something that we would find in any of the organisms that are prior to this point. If we talk about cladistics and the problems with cladistics, the problems are very similar to what we have with the phylogenetic trees. One of the biggest problems is time. These branch points don't have a set time on them, so it's hard to get a real idea of when something evolved with respect to the geological record. What we can do is we can try to compare it with the geological record. We can try to correlate the length of a branch with DNA changes that perhaps have taken place. So if we have a gene that we can use as a molecular clock, we can try to put something together similar to what we see here. And what this means is that we can see that right here, say the human and the mouse, those diverged fairly recently compared to the other organisms shown on this particular cladogram. When the cladograms are put together, the idea of parsimony is very important. And what parsimony means is that the simplest idea is usually correct. So this means that the tree that would involve the least number of evolutionary changes is most likely to be the correct evolutionary tree. If we look at two different examples here, we have a hypothesis tree number 1 and we have hypothesis tree number 2. And the little red marks that you see here, those indicate the number of evolutionary changes that would have to take place for each of those trees. And this is with us comparing the shared derived characteristics. So in this first tree, hypothesis number 1, if you count the number of little red lines, we've got 6 total evolutionary changes that would have had to take place. If you look at hypothesis number 2 and you count the red lines, we've got 7 evolutionary changes that would have had to take place. So what that means is that if we're using this concept of parsimony, then hypothesis 1 is most likely to be the true one, because that would be the simplest idea or the simplest tree that we can come up with. It involves less changes taking place. Here's another example. We've got 3 species. We're looking at some differences between them, and the first one that we see here, we would only need 1 evolutionary change to take place. But if we were to go with the second tree here, we would need that 4-chamber heart to evolve 2 times, separately 2 times. So that's going to be a lot less likely to happen. So what that means is that this first tree is the one that we're going to go with, because that is the simplest explanation or the most likely tree that would have taken place. So if we take this idea of cladistics and phylogenetic trees, this is what we have for all of the organisms that we know of. So this is our typical tree of life. In this tree of life, notice that where this little blue dot is, that's where we have our common ancestor for all of life. So when we talked about how life evolved, that's where it's thought to have taken place. We then have 3 major groups of organisms, which are now called domains. We have bacteria, archaea, and eukarya, and all organisms are going to fall into one of those. Throughout the rest of this semester, we're going to talk about those 3 domains, and we'll talk about the organisms that make them up. But notice when we look at this, that eukarya and archaea are going to be more closely related to one another because they have this common ancestor here. So they will be more closely related to one another than they are to the bacteria. What we're seeing on this slide is a ring of life as opposed to a tree of life. This ring of life is how a lot of scientists view the evolutionary history. And what this means is that rather than have one common ancestor that then diverged into archaea and bacteria and eukarya, that we might have had a combination of archaea and bacteria that actually fused together and gave rise to eukarya. So that's a little harder to draw if we're talking about a tree structure.